So will we be able to see all of everybody?
I think, no, I think we're going to shut off the video, right?
Yeah, yeah.
Oh, so we won't have a gallery view of everybody who's watching?
Mm-mm.
I'm going to run downstairs and get a glass of water.
Okay, we have some already. Can I open that up? Are they not in? Oh, attendees. Yeah, we got, so we can watch them as they come in. I was gonna type that. I can't type chat. Oh, that was the Q&A. I was just going to send it to the attendees, but it's not working.
Erica and David, can you see who's been signing on?
Yep, we've got 4, because 2 signed on together, 5.
Oh, I see.
We've got a few.
I'm seeing.
They're coming.
Okay, cool.
Coming in. So Kristen, with the Q&A, since I'm a panelist, can I not type? Not that I need to, but I was just playing with it and it doesn't look like there's like a box for me to type in. Is that the case for panelists?
It's a great question.
Could you go to the chat?
Yeah, I can type in the chat. I can type to all panelists or panelists and attendees, but I was just curious if with the Q&A, I want to make sure, if questions are being answered in the right spot.
Yeah, so I think we can only utilize the chat, but you'll be able to hop in and answer questions and stuff as questions roll in. But if you have specific questions, you can do it through the chat
So the attendees will be able to access the Q&A? Is that?
Yep. Mm-hmm.
Okay, I just wanted to make sure of that, that's all.
Yep.
Second message.
Oh, I know what I figured out. Better. I don't know if you have this issue, David, but I find it really annoying when I see my own reflection in my glasses on the Zoom calls. And I found a pair of glasses that doesn't do that as badly. Erica, are most of the folks here going to be in Baltimore?
It's gonna be a good number in Baltimore, but did reach out to some of the Ewing sarcoma groups, and so I know several of them registered, so they could be anywhere. I don't know. Yeah, I mean, we reached out to our fan, the Facebook fans and stuff like that too, so they're all over also. So. Glad we're getting good attendance. Oh, got somebody in California. reported, who commented in the chat box. Yeah, there's a few, because I didn't put in Eastern time. I didn't even think about it. So people were asking what time zone. We've got North Carolina. Yep, because we're not muted.
Oh, I see Chris Carson. I know Chris.
I know him from the Ewing Sarcoma group.
That's a familiar name. Yeah. Love to hear how you guys are doing at some point.
She emailed me. Got a couple more just now.
Okay.
Mm-hmm.
I guess they automatically get the links, right?
Yep, that's why I did it that way. Oh yeah. That's this one right here.
So what if we go till 7:35 and then start? Does that sound good to you, Erica?
Works for me.
Okay. So for everybody who got here on time, you shouldn't be penalized for being on time.
I am with you there. I
Spoken like a true school teacher, right?
Pet peeve of mine, yep.
Yeah, these things always take a little bit of time to get people in, because of the nature of this, but I think 5 minutes is plenty, so that'll be good. Okay. Probably time to get started.
Now Sharon's gonna start barking, of course.
Yeah.
We're ready whenever you are.
Sure.
Hey, why don't you go ahead?
All right, thanks everybody for joining us with Dr. Toretsky, and this is Kristen. Christy, you go by Christy? Christy. They're from Georgetown University, and we appreciate everybody coming in to hear what Dr. Toretsky is doing with his research. This is possible because of all of you generous donors and supporters of Ari's Bears, and we hope this is not the only grant we get to give out. So we're happy to be able to have this and have everybody joining us. Thank you.
Great, thank you so much. So as Erica said, my name's Christy Augenblick. I work at Georgetown Lombardi with Dr. Toretsky and others to help secure funding like the grant we got, amazing grant we got from Ari's Bears for amazing research and clinical care at Georgetown. So just so everyone knows, we're going to be using either the chat or the Q&A function, whichever is easiest for you to answer questions. Feel free to do that as we go. We'll try to get to as much as we can and make sure Dr. Toretsky is seeing those questions as they come in. So Dr. Toretsky is our presenter today. He has been with Georgetown Lombardi since 2002 and was named Chief of Pediatric Hematology Oncology Division in 2017. Under his leadership, Dr. Toretsky has transformed the division and expanded it to include an adolescent and young adult department. Dr. Toretsky actively pursues research that will lead to new and more specific therapies for a rare cancer that many of you are probably aware of, Ewing sarcoma. Ewing sarcoma tumors contain a unique target that is not found in non-tumor cells. This unique target offers an opportunity to create new medicines that will more specifically eliminate tumor growth while sparing normal cells, as I'm taking all of his thunder. This generous grant, again, from Ari's Bears will help fund this important research that Dr. Toretsky is doing through Georgetown and also his work with his company that he co-founded, Nocturnal Therapeutics. They currently have a clinical trial that's going on right now, which we're definitely going to hear more about. So without further ado, I will pass it on over to Dr. Toretsky.
Well, thank you, Christy, and thank you, David and Erica, for your generous contribution and everybody who's contributed to that through Ari's Bears, and also to all of you who've gotten on this call tonight. We're certainly living in a weird world. This is an incredibly weird way to do this. It would have been much better to meet in person and have a hug and a cocktail and get to do this. But, well, we'll do what we can do. Everything is a little bit different. So I'm going to get rid of this crazy background, and I'm going to go to this and make sure hopefully it'll still work. Okay, first slide. Yeah, everybody see that? Erica, we're good? Okay, and you guys can hear me okay?
Yes, we're good.
Volume is okay? Okay, so, so this talk is one that I've modified specifically tonight for Ari's Bears, but the title of the talk is one I've used, From Idea on a Napkin to Clinical Trial, Creating a Drug for a Rare Cancer. And a longer version of this talk, I've often talked about a lot of the different hurdles we've had to overcome. Today, we're going to keep it more streamlined. I'm going to shoot for about 30 minutes. So that there's plenty of time for questions in the end. If you want to hear a longer version with a lot more scientific detail, I've been fortunate to be asked by the Bone Cancer Research Trust from United Kingdom to give a keynote on October 16th. I don't think that they're charging registration anymore for their meeting, and you don't have to travel to London to hear the talk. So if anybody's interested in that, you would be more than welcome. You'll hear some of this again, but there'll be a little more depth of science. So one of the questions I often get very early on when people meet me and they know I have an interest in Ewing sarcoma is they ask, how did I decide to study Ewing sarcoma? A disease that afflicts a relatively small number of people is something that I've had a passion for almost 30 years now, 29 years. And there are two things that are interesting coincidences. And I'll be honest, I did not have either of these things happen. Well, perhaps the second one, but the first one clearly not. When I was a junior at the University of Wisconsin, I was doing research and I was working with a pediatric oncologist named Jonathan Finlay, who, while I was doing research in the lab, he would have me along to clinic, and I met a patient who happened to have Ewing sarcoma, and he was a freshman. And I would often work in the lab in the evenings. Sometimes I'd order a pizza, look for somebody to share it with, and he was a freshman, so we hadn't had a pizza. And sadly, he died halfway through his freshman year of Ewing sarcoma. And honestly, I didn't say at that point, I'm going to go out there and cure this disease, but I've looked back on that as a really interesting event in my life. And there's more, but for that's good for now. And then in 1991, I started my fellowship at the NCI. And at the time, there were some Mesna, which you probably know about if you've been looking after Ewing sarcoma patients, is an agent that allows us to give certain drugs as kind of an antidote. And that was only available at the NCI in the 1980s and early 90s. And so there were a lot of Ewing sarcoma patients that wanted to get that drug. And so we had a fairly significant population of mostly patients with Ewing sarcoma who had metastatic disease at diagnosis. And as you know, that's a very challenging group to treat even today. And I had a lot of them in my cohort of patients, and sadly, they all died. And it was right around that time that a very significant event happened in the world of Ewing sarcoma, which is that the chromosomal translocation that we'll talk about in a little bit was first identified. So most of you on this call are familiar with Ewing sarcoma, but I just want to point out that Ewing sarcoma is this really unusual cancer that really can occur anywhere in the body. And here's a couple examples of it occurring in bone. Dr. Ewing, who described this cancer back in the 1920s, separated it from other bone cancers because it had radiation sensitivity. Now here's a slide that I show that Ewing's can occur anywhere from the top of the head in this CT scan to the tip of the finger. Sadly, patients with metastatic disease don't do very well. Patients with localized disease do better, but it was clear to me from a very early time in my work with patients that we had to come up with something to improve their survival. So this is the key. In the early 1980s, the chromosomes 11 and 22, shown here in what's called a G-banded karyotype, were identified to undergo something called a translocation. And so basically that means a little bit of 11 breaks off and goes to 22, and a little bit of 22 breaks off and goes to 11. Think of this, for those of you old enough to know this, the Reese's peanut butter cup ad, he got chocolate in my peanut butter, she got peanut butter in my chocolate. And somehow this change, this break, leads to a new gene that is translated for a protein we call EWS-FLI1. And this talk is all going to be about, how we were going to figure out how to turn off EWS-FLI1 in Ewing sarcoma. This diagram, this picture is a cell, and it shows what happens in cells with EWS-FLI. Where we go here? The green dot here is a piece of chromosome 22 from the non-translocated, so the normal chromosome 22 shown here. The red dot is from the chromosome 11 shown here. And when some of number 11 goes on to number 22, the red merges with the green and gives a yellow dot, which is what you see here. And this is the way that a lot of Ewing sarcomas are diagnosed today. This is called a FISH assay, fluorescence in situ hybridization. Now, we've known for a long time that if we could get rid of EWS-FLI, and this example is using something called siRNA. I won't have time to explain that today, but just let me tell you that in both groups of mice that were implanted with Ewing sarcomas, this group of mice received a small piece of RNA that caused EWS-FLI to go away. And this group of mice got a control that didn't have the same sequence, so it didn't go away. And you can see that in these mice, these colored spots show the active Ewing sarcoma tumors through a technique called chemiluminescence. And here's one that even metastasized to the lungs. And these mice with the siRNA were cured of their Ewing sarcoma. And so this was a great trick. It works in mice, but this was never something that has even today been able to be used in patients with Ewing sarcoma. So we had to figure out a way to be able to get rid of or turn off EWS fly without using one of these siRNA type tricks. Okay, next slide. So one of the challenges in cancer overall, and I put this slide up because I wanted to make a point that cancer cells and healthy cells don't look that different under the microscope. Cancer cells are not blue, healthy cells are not pink. But one of the challenges in cancer biology is to identify those things about cancer that are not found in non-cancer cells, and therefore you might have a good target to develop a drug against. And in our case, EWS fly is one such target. And a good target will improve the therapeutic index of a drug. And what is a therapeutic index? The therapeutic index is the amount that kills the patient divided by the amount that kills the tumor. So ideally, you'd like to have a drug that has to be given at 100 times the dose to harm a patient compared to a single one time to kill the tumor. So in other words, if it took 1 milligram to kill the tumor, it would take 100 milligrams to harm or kill the patient. That would be an ideal therapeutic index. But unfortunately, most of our drugs today are in this 1.5 to 3 range, meaning that we get a lot of side effects and a lot of collateral damage as a result. And one of my goals in setting out to do this project was not only to improve the survival of patients with Ewing sarcoma, but I wanted to give and provide the cancer community with something that would be better targeted. Now, I wasn't the first one to think about targeting cancer. It's been around for a long time. This is a picture of Brian Drucker, and Brian was responsible for bringing a drug called imatinib for chronic myeloid leukemia into clinical trial. Brian is now the Cancer Center Director at Oregon Health Science, but he basically was a fellow in Boston and a young faculty member. And at the time, he wanted to think about how to turn off the gene BCR-ABL that causes chronic myeloid leukemia. And he came up with this drug, imatinib. And it took a long time for people to really understand that this was possible. But in the early 2000s, clinical trials were shown to be very effective, and this becomes a very effective treatment. But it also became a harbinger of treatments to come for many cancers where you could turn off similar genes called tyrosine kinases. However, in Ewing sarcoma, we don't have a kinase. The EWS fly protein is actually a transcription factor. And even though transcription had been recognized as a driving force of cancer for a long time, basically what people had been told ever since I was a fellow in the early 90s was that you could never target a transcription factor because a series of scientific reasons. Okay, so when we started this work, thinking about how to turn off EWS fly, we had to use a significant amount of imagination. So let me give you my little imagination quiz, if I might. And I can't see your faces, which is really a bummer because I love watching people's reaction to this. But I would like you to imagine a color you have never seen before. So just think for a second. Our job was to turn off EWS fly. We thought about that over 20 years ago. There was no paradigm on how to do it. And we had to think about how to do it completely in a world that said you can't do it. And I love doing things. You can ask anybody who knows me that people say you can't do. And I want you to know, you've had a minute, imagine a color you've never seen before. So that color may be here, but it may not be. And if it's not, you understand the challenge we were facing. So this was our paradigm. And I hope this isn't too overly simple, but we'll make this one step more complicated in a minute. So here's what we envisioned. And this was about 20 years ago we thought about this. EWS fly would be the center of a sort of chemical reaction that occurred in cells. And it included its ability to turn genes on and turn genes off through this part called fly1. And we knew that in doing that, that ability to turn genes on and turn genes off, it had to do it with other proteins. It had to interact with other proteins. One of the proteins we identified very early on that literally interacted with EWS fly was called RNA helicase A, or RHA, fits better in the blue box. And we had this idea that if we could disrupt an interaction of a protein with EWS fly that was needed for cancer cell survival, we could create a new drug. Now the trick would be, remember the thing about therapeutic index, we had to think about how to do this from the EWS fly side, leaving the RNA helicase A protein, which is found in all normal cells, alone. How did we do that? So we thought about how a new drug could actually interfere with this by sticking to EWS fly and blocking RHA, okay? And it would look something like this, where we created a small molecule that sticks to EWS fly, but doesn't stick to RHA, but blocks the spot on EWS fly where RHA binds. And at this point, I'd be looking at you all, hoping that you're all nodding your heads saying, yeah, I get it. So hopefully you get it. I can't see any questions coming up, but I'm gonna pause for a second and ask Christy if there's anything in the question box so far.
Nope, you're looking good.
Okay, so I'm assuming everybody's nodding that you get it so far. So let me show you a little bit more detail of the science. Because I love showing people the details of the science, and I hope that brings you along so that you can understand that, you know, my simplified drawing is simple, but there's a lot more that I would love to share about this. Okay, so this is probably unfamiliar to most of you, but I'm going to walk you through it step by step. So starting at the bottom of the slide, you have our EWS fly protein, and here's a piece of RHA, not the whole thing, but a small piece of it. And it happens to be, in case you're curious what these numbers mean, 630 through 1020 are the amino acids, this 400 or so base piece, or 400 amino acid piece, was attached to another protein we call GST. And we knew that RHA would stick to EWS fly. And the way we could determine and prove that to ourselves was we could take EWS fly when it's floating in a tube and use an antibody, and this Prot-G allows us literally to centrifuge EWS fly to the bottom of a tube, okay? And if RHA was present by attaching itself to EWS fly, we would see both proteins present, and that is shown here. So when we look for EWS fly with an antibody, we can see the spot, and this is the GST spot. And this tells us in lane 3 that both RHA and the GST RHA and the EWS fly were together, just like you see in the bottom of the slide. Okay, now what we did is we added in a small piece of RHA, which was a mimic. So it's just a few amino acids, 10 to be exact, a mimic of the section of RHA that sticks to EWS fly, such that the mimic sticks to EWS fly and blocks RHA. And you can see that here. So we add in 10 micromolar of the mimic. And you can see that we now still get EWS fly, but we no longer get the RHA band. And one of the ways scientists prove things to each other is instead of just using one concentration, we use a range of concentrations. So as we use less of the peptide, you can see that the interaction with RHA returns. And so that was a proof that we could take this little tiny peptide, block the two proteins, and at least show in a test tube that we could keep these two proteins apart. And that was fundamental to our getting to the next step. So what we did, and I apologize because I don't think this cartoon is going to run, but what we did was we took EWS fly and we put it on a surface, this Biocore biosensor surface. And it turns out that we literally ran chemicals one by one over the surface. And when one of the chemicals stuck, we would get a signal on the graph telling us, aha, there's a chemical that just stuck to EWS fly. that's one you want to put in your consideration box. Okay, so here, again, chemicals were floated over EWS fly, and when one stuck, we would get a signal. And this is an example of one of the chemicals that stuck. And what we did is this chemical was pretty good, but we wanted to say, could we make it better? And what this slide shows is a chemical reaction where we could change this end of the molecule. We call that substitution with all different things. And this particular substitution, you see I've gone from a Cl, which is a chlorine atom, if you remember your chemistry, to an OCH3, which we call methoxy. And it turns out that that particular molecule, actually we'll mention real quick, that particular molecule comes in two flavors. And it's because of this spot right here. And one of the things that showed us was there were two flavors of the molecule. And it turns out that only one flavor, the S version, we call that an enantiomer, one version, the S version, was active in killing Ewing sarcoma cells. And you can see this is a growth curve showing how the drug, as you increase the concentration, kills them at very low concentrations. Here's a mixture of the left of the S and the R, and here's the R not killing them. And what we did is we did this over and over again. Here's an example. I showed you already how when the two proteins interact, you see both spots. Now we add in the YK4279 and we disrupt it. And if we do it with the S, we disrupt it. We don't disrupt it with the R. Here's another assay showing that we can activate genes with EWS fly, and we can turn that off with the S or the racemic, which is both together. We don't do it with the R. And oopsie. And this is a splicing assay where we do the same thing. And again, the R is equivalent, oops, there we go. The R is equivalent to doing nothing. And there are a number of papers here that I cite. If anybody's interested in those, I'm happy to send you the reprints from those papers. If I hadn't mentioned it already, this has really been, a 20-year odyssey. It was really 16 years from the time we came up with the idea on the napkin to the first patient going on the clinical trial. Okay, so once we knew that this compound kept the proteins apart, then we went to do experiments in animals that had Ewing sarcoma tumors. And you may have heard this term apoptosis. Apoptosis means a kind of special cell killing. And it's special because it's irreversible. Once it starts, the cells are going to die. So that's a good thing for a drug to do, to cause apoptosis. And these are 4 different tumors from mice treated with Ewing sarcoma. They're all the same tumor from 4 different mice. One was treated with nothing. One was treated with the R enantiomer, which I've already shown you was inactive. And you can see there's no difference. These are cells that are totally happy and living. But when we treat with the racemic or the S, you see these brown spots. And the brown spots are cells that are undergoing apoptosis. You can almost tell at this magnification that the nucleuses are kind of falling apart, little spots falling apart. That's a marker of apoptosis. And these cells are irreversibly dying. So then we went ahead and we took our drug, and I'm happy to answer questions about this if people are interested, but because of challenges in doing the mice, because of the solubility of the drug, we went ahead and we did an experiment in rats. The reason this experiment was possible is because we wanted to be able to give the drug by continuous infusion. And you can't put a catheter in a mouse, but you can put a catheter in a rat and give a drug just like if you gave an IV to a human. And if you look at the y-axis, notice the scale. This is centimeter cubed. So 1 centimeter cube is, if you take your forefinger and thumb and put them together and make a little loop like that, the size of a large pea, like a garbanzo bean, that's about 1 centimeter cubed. And you can see here, we put these tumors in the leg of a rat, and we started at 2.5 to 3 cubic centimeters. And this was a very expensive experiment to do, and I can, again, talk about that more if people are interested. But we had 6 rats getting the drug and 3 control rats. And The experiment cost in the end about $350,000 because of the care and how we had to do it. And you can see that all three of the control rat tumors took off and grew pretty quickly. 20 cubic centimeters is pretty big. That's about the size of a golf ball. And the 6 rats that were treated, 1 rat chewed through its catheter early, and then that rat didn't get drug anymore. And then of the 5 remaining, 3 of the rats had absolutely no change in their tumor over almost a month, and 2 of the rats had complete regressions. In fact, it was so complete that when we did a necropsy and looked at the leg from those animals, you can see here in the control, this is what Ewing sarcoma looks like under the microscope. This is from one of the rats from the control rat. And CD99 is a stain that we use to identify Ewing sarcoma cells. You can see all these golden ringlets around the cells. This is a very good example of what Ewing sarcoma looks like under the microscope. And you can see in the leg of the S-treated mouse with the active drug that there's a lot of spots like this. This is necrosis, and there's some fibrous tissue here, but in the CD99, you don't see any positive cells like this. So we had done a complete microscopic cure of these mice. So this basically led us on to a clinical trial. Now, from the time we did this experiment to the time we did the clinical trial, there were 2 companies that I founded that didn't succeed. And finally, in 2013, after a significant amount of attempts to get this into the commercial space, Scott Glenn reached out to me. Scott was a serial entrepreneur from San Diego who was being asked to look at some other projects, and he found our work, and he decided that he was going to found a company around our ideas. And with that, a company called Tokelis was born in 2014. And what Tokelis did was create a series of additional chemicals. One of them they called TK216. And they also did all of the necessary preclinical pharmacology. So how does the drug get distributed in the body? How does the drug get cleared in the body? What are the potential side effects that could happen when we would put this into a human for the first time? And all of the regulatory work to the FDA that one needs to do in order to begin a human clinical trial with what was called a first-in-human new chemical entity. So this drug and anything like it had never been given to a human before. So there's 2 things about that that were fascinating to me. One of them is that this Scott and his team really knew how to work with the FDA, because in December of 2015, they applied for their investigational new drug application, which is what one needs in order to start a human clinical trial. And they sent that application in around December 18th, so just before the holidays. And by January 9th, we had gotten approval. So I want to stress how interesting and amazing that is. It was less than 30 days from the time the company asked to the time this new drug application was granted. And it was for 2 reasons. And this was over the holidays, is because the folks on this team really knew what they were doing, and it led to a very, very quick approval. So what did that get us started with? Well, it turns out that got us to cohort number 1. Now, cohort number 1 was a dose of the drug that was very, very, very low. In fact, it was 1,000 times less than we would expect the result in a human to be effective. And that's because safety first. We needed to start at a very low dose, and that's how the FDA asked us to start for 2 reasons. One, because by the second cohort, we had asked to go from an age 18 down to age 12. And that had never been done for a small molecule like this before. And so the FDA approved it. They said, if you put 3 patients on at this dose and nobody has any serious problems, we'll let you drop the age to 12 so that you can enroll younger patients for the second dose level at 36. And what happens in a phase 1 clinical trial is you double the dose until you start to see any side effects. Now, we knew that until we got up to around this level, we probably weren't going to see an effect, because we knew from the preclinical studies where we needed to go. But one can always be surprised. And it's really something that people don't have a good way around in clinical trials, because at some point, if we had started at this level and we had 3 patients get sick really badly right away, it would have killed the program and we would have never known where we could go with a safe dose. And so this is how the FDA requires you do it. And finally, the other thing we knew from the rat study was we wanted to give the drug for as long a period of time as possible. And so eventually, we went through a series of dose levels, and I'm showing you the schematic here. We got to this cohort 9, where we got to 200 milligrams per meter squared for 14 days and no dose limiting toxicities. You can see here we extended from cohort 8 to number 9 of the infusion by 4 days. And this became the dose that we were going to use to go forward. There were 4 patients enrolled on this dose. One of the patients enrolled on this dose was enrolled in March of 2019 after having multiple relapses of Ewing sarcoma, at least 4, and had been on 4 different regimens over 2 years, and had originally presented with metastatic disease, which, as you remember at the beginning, is really a real challenge clinically. I'm proud to tell you that this patient remains in complete remission now 18 months later. And there's a second patient who's also in complete remission. And of 15 evaluable patients from this expansion cohort, 5 of them have stable disease and remain on drug. And there are 2 complete remissions for an overall response rate of 47%. So for a drug so early on in clinical trial to have that kind of response rate, we're very, very excited, and hopefully that will be a harbinger. And the sites that are now doing the clinical trial are now expanding, so we'll be able to continue to see what our response rate looks like, and hopefully get this included in future clinical trials upfront for patients with metastatic disease. And there's discussions happening now with COG to start to think about that. But one thing I want to point out is that even though our initial goal was to have a drug that would inhibit EWS-Fly specifically, it turns out the drug is probably working through part of the Fly1 part of the fusion protein. And one of the things we've found along the way is that this drug could have particular efficacy in myeloid leukemia and lymphoma. And we published a paper with a collaborator in Switzerland last year that talks about that. And we're planning clinical trials to go forward and do that. In the meantime, on the laboratory side, we continue to do experiments to understand more and more about how EWS-Fly really works in cells, and to start to think about the kind of potential resistance that could happen to patients who aren't responding. You know, the question is, why aren't some patients responding? And we're trying to figure that out at the molecular level. So at that point, I want to just point out that this work was done by an incredible team of people. Aykut Eren was a partner of mine since 1999, when he joined me as a fellow, and now he's a full professor, and we continue to work together 20 years later at Georgetown. There were bioinformaticians, biochemists, chemists, many, many people involved in this. There's a picture of Dr. Eren there. This is us. I believe this was the Billy Goat Trail. We tend to take the lab out hiking every year. But I want to make a particular point in my thank you slide to thank Erica, David, and all of the people from this call and their friends who made Ari's Bears Foundation a success. It's really wonderful that you're able to do this. And as you can see, the work that we've done so far required a significant amount of support from a number of different groups. We were a beneficiary of money from the Recovery Act and Children's Cancer Foundation of Baltimore, Maryland, actually now in Columbia, but it's the same foundation. That organization was run by literally a little old lady out of her house, Shirley Howard, of blessed memory. And she provided the seed money that really let us get those initial experiments going that led to the drug. So it took all of that before the NIH kind of kicked in their part. And groups like Ari's Bears have been critical in helping us do small parts of the puzzle so that we could get to where we are. So with that, I'm going to stop sharing. And I'm going to turn off this crazy background I have.
All right, we'll get my cool occupational therapy background here.
and you'll see my boring home office. And I would love to take any questions, and I'm totally happy to have people unmute if that's possible, and they can ask them in person and introduce themselves. Or if they want to just put them in the chat, that's fine too.
I just want to say thank you before, hopefully people, I don't see any questions being typed, but thank you for doing this. Thank you for your research. I know Dr. Abalafia had mentioned you when Ariella relapsed. She would have been too young, but it was definitely one of the, trials we were told to consider. And so, it sounds like it's doing good things.
I want to say something to that, Erica and David. My heart goes out to you guys. One of the things that I told the folks at the company was it was critical to me to bring the age as low as possible, as soon as possible. For the expansion cohort, we're down to age 10. So we can give this to patients as young as 10. I'd love to have had it go lower, but with regulations what they are, it's kind of hard to do some of those things. But one of the things that was particularly sad to me is that there have been people that have joined this journey along the way. They've become known to me by reaching out. And some of them haven't survived their cancer journey with Ewing sarcoma, like Ariella. And they're people that knew that this was coming along, were encouraging to me, very encouraging. I'm thinking of one young man who was already a father, and he was a biochemist himself. And he said, I know I won't live long enough for this to get to a clinical trial, but, I'm just, glad to know that you're doing the work. So it's been really hard along the way. Yeah, it's been hard to not, but now that we're here, but it's been hard along the way because there's a long period of time where we didn't know how we'd get this into the clinic.
You got some questions?
That's a whole other, those failed companies, boy, that's a beer. We gotta have a drink over that. You would not believe the stuff that we went through. Let's see, are there questions here?
There are some questions in the chat. Do you want me to read them to you, or you want to read them?
No, actually, it looks like the first one is from Karen.
Yes.
It says, what are the parameters to become part of the trial? Active disease, size. Can a child older than 18 be on the trial? Yeah, actually, the first part of the question is you have to have measurable disease because it's a phase 2 study right now. We're looking for efficacy. So we have to be able to measure disease shrinkage. So you actually have to have it could be a centimeter, but you have to have something that we can then measure after starting the drug to see if it's shrinking to know whether the drug is working. And we're working on developing, and this will be probably something that I use this gift from Ari's Bears for, we're working on these pharmacodynamic assays, meaning can we find ways to measure how the drug will work without actually having to keep taking pieces of tumor. And so we're working on that. I think the oldest patient that's been on the study was in their 70s. So it turns out Ewing sarcoma affects patients well into their later years. And there was one patient who was a breast cancer survivor who got Ewing sarcoma as a second malignancy, who went on the drug. So older is no problem. And I'll have to tell you that she tolerated the drug really, really well. I can't remember where she fell out in terms of which stage or where she was at. But a couple of the patients who are doing really well now are actually older patients, and some are really young. Let's see. For the 2 patients in complete remission, are there comments, biomarkers? Yeah, so we're working on that. That's exactly what we're trying to figure out. So actually, as I've been working on, as I put this presentation together, there is a group of people that I'm working on to create what's called a program project. A program project grant is an NIH grant that brings together 3 otherwise very large NIH projects into one larger project that allows them to work together. And Octernal, the company that is sponsoring the trial, has agreed to provide us with patient materials that were on the trial, where we've collected them as they've gone along to start to ask those questions. And that program project will be reviewed by NIH program officers literally 2 weeks from today. And if they agree to allow us to go forward, we'll write an application due in January, and that will allow us to go forward with an incredible group of investigators from literally from LA to Toronto to try to solve that challenge about what are some things that will tell us who will respond, who won't, and how we can get around that. Yeah, Chris Carson writes, is it possible to find a biomarker? Yeah, that's exactly what we're working on. Chris, we can, yeah, that's the goal. It's very challenging, as you know, because, you know, people who have Ewing sarcoma, especially once they've relapsed, the thought of having more needles stuck into them is not appealing. And we're trying to find ways to use blood as a surrogate for sticking more needles in our patients. And how many active participants from Portia Wright? How many active participants are currently enrolled? And is it, well, yeah, we're now technically into phase 2. There are 15 evaluable patients, and we've now opened up the cohort. There's an amendment that's gone through for about another 20 patients. So we'll be able to complete a full phase 2 study now from the company. The company, by the way, I forgot to put my disclosure slide. It's called Oncternal Therapeutics, ONCTERNAL. I'll put it in the chat. And you can Google them and find them online. And just so you know, I'm a shareholder and a founder, so there's my disclosure. They're working hard to raise money to get us through to the finish line. Their goal is to get this drug approved. And I've been really, really proud of the team there because they've stuck with this. It took a long time to really get the clinical trial rolling. And after that first patient started responding, the patient recruitment just took off. So it's been really good from that perspective. Let me see. A question from the Q&A from Diane Randall in the San Francisco. My son is 26, second relapse. Does this drug overcome chemo resistance? Well, all I can say to Diane is that the patient who's been on this for 18 months had 4 prior regimens. So clearly they were resistant to something. They kept getting disease back. Ewing's is kind of weird with chemo resistance. People do tend to respond over and over again. I would say if you're in San Francisco, you can go to the company website or you can go to clinicaltrials.gov and you can see where all the different sites are. And I would say that the nearest sites to San Francisco is either LA with Noah Fetterman or Portland with Laura, I just spaced on her last name. But the clinical trial is on the West Coast, both in LA at UCLA and in Portland. And either one of those would work from San Francisco, I think. If you wanted to fly or go somewhere else, there's sites all over the country, but those are the 2 closest ones. Also, I don't make particular recommendations for one site or another, and I try to keep arm's length from any patients getting enrolled on the study because of my conflicts with Georgetown and everything. I have to be honest about that, and I want to be careful not to show any sense of conflict. What are the known side effects from Barry Patnoy? Yeah, it turns out it's really interesting. Patients have tolerated this drug extremely well. The only real side effect that we've seen repeatedly is low white blood counts. And for Ewing sarcoma patients, a low white blood count is nothing. We give GCSF, their white count comes back up. It turns out that the fly-1, which is where we think the drug is attacking an EWS fly, is also one of the critical factors needed to make white blood cells at a very early stage. And so we think that neutropenia, that white blood cell effect, is actually an on-target effect for one of the cells in the body that, of course, we tried to avoid by creating this drug against EWS fly, but that was a trick. So we're continuing to work on new drugs that may do that, but that's a real challenge. Erica wrote, can participants who have already started chemo participate? Unfortunately, you have to have recurrent disease. So you have to have, we don't have it open right now for patients who are getting upfront therapy. Because again, we don't want to do any harm. So ideally, if patients, we don't want you to relapse, but one of the things that that I feel fairly comfortable with now is if patients relapse the first time back before we knew the drug had any efficacy at all, we would be reluctant to recommend going on this clinical trial the first time you relapse. But based on what I've seen now, and we have it a couple of patients, and they've done kind of pretty good actually, go on the drug with their first relapse. And as long as there isn't a life-threatening nodule or something that needs to be treated immediately, it's something that the patients can do if they want to. They don't have to have multiple regimens. They can go on for a first regimen, and that might actually make it easier for the drug to work, but we don't know that. We're still figuring it out. Chris Carson writes a long paragraph. Let me start. How can we improve the efficacy of what you've already created? Are you looking into additional combinations? Are you considering additional strategies to your current combinations with other researchers? How can we increase the overall response rate? How can we take your success and build on them and create an improved trial for current relapse patients? Appreciate all the, okay. So let me kind of take that apart a little bit. We are looking at combinations. In fact, we published a paper in Science Signaling in 2017 showing that vincristine is very synergistic. And the nice thing about vincristine is that the doses we use it in Ewing sarcoma, it's not very white blood count suppressive. So we don't really suppress white blood cells with vincristine, so it's a good synergy drug for TK216. There are other possible combinations. We're actually working on a manuscript now where we've looked at a number of those, but it's hard to get multiple ones in clinical trials at one time. Yes, Chris, that's something that we're thinking about, and it would be a great idea. We've even done some combinations with other experimental agents, and I think that for now, the company doesn't have the bandwidth to start testing multiple other agents. But if they got an approval with TK216, then the possibility of building other experimental drugs in would be possible, because now you'd have one approved drug and one experimental drug. So it could be done that way. And there's a number of other drugs that are in the pipeline that are being created by my colleagues, either LSD1 inhibitors or trabectedin analogs that Patrick Grohauer is making, or Steve Lesnick's LSD1. So there's a number of things that are coming along. There's also some of the multi-tyrosine kinase inhibitors are useful for Ewing's, but they have a lot of side effects and they don't work for very long. But some do. There are a couple of long-term things. I think that answers all of Chris's sub-questions. And next, Diane again. It's sort of more of a direct clinical question, Diane, so I'm going to pass on that for now. It has to do, I'll just say broadly, with whether surgery is appropriate at different times, and I can't really answer that in this context. It's not, without scans or anything, it wouldn't be really appropriate. Maintenance arm, Piero Spada, I hope I pronounced that correctly. Any talk of maintenance arm for the trial in a later phase? Yeah, that's an interesting question. And this patient who's been on the drug for 18 months without a relapse, that's scary, right? When do you stop? How long do we keep doing this? And he's tolerated the drug extremely well. So we're trying to think about what that would look like. We don't really know. And so Hillary Lang asked the question, is there talk of adding maintenance? It used to be that maintenance was out of the question for solid tumors, but rhabdomyosarcoma sarcoma is now talking about and has a trial going with maintenance. And Ewing sarcoma, I was just part of the COG meetings. It is on the table. So people are thinking about it, but we'd have to know what are we going to measure to know whether it's working, or are we just going to give somebody more drugs? So it's hard to answer that. It's a tricky trial to design. And Piero Spada asked, how long would a patient stay on this if they're responding? Well, I think I sort of answered that. The answer is we don't know. We'll keep them on it. The company's got a lot of drug. We'll keep you on it as long as we can if it's working. And Diane writes, how much time is required at the treatment site? Okay, so this is an interesting problem. So like I said, in the rats, we had to give this as a continuous infusion, and we made a decision at the company that I pushed very strongly for at the very beginning, which is that we would give this to humans as a continuous infusion. My knowledge of Ewing's was that virtually everybody that relapsed had a catheter. And if you have a catheter, you can get a hip pack, and the drug is a small bag, and you can get the drug continuously. And the reason is if the drug would have variable absorption from the stomach of different patients, we never really know how the drug was working, because we wouldn't know how it was being absorbed at any given time based on what somebody was or wasn't eating. So we made a decision up front, since we had to get a proof of principle to do this as an IV drug. We're currently working on oral formulations. In fact, there's a grant pending at the NIH for doing an oral formulation. And so that's where we're at. So currently, patients do have to be at the treatment center for periods of time. Different treatment centers have worked it out. And in some cases, somebody will start at a treatment center, and then we will approve, the company will open up a site at a home center if somebody's on the drug, so the drug can be given closer to home. We had that happen in a couple of instances. One patient was coming from the middle of the country to Duke, and we opened up a site in the middle of the country so that they could get it close to home, and that worked out really, really well. So the company is really willing to try to do those things, but initially you have to spend a couple of weeks at the site, or at least come back and forth. It's going to be going from 4 days to 7 days soon, so that's a good thing. Specific criteria, yes, there are specific criteria. I don't have them all memorized, so I'm not going to say them now, but the key thing is you have to have some measurable disease, and you have to be reasonably healthy. Question from Hillary Ling. I'm hearing about GI issues. Is this one of the main side effects? There have been a couple of patients with GI issues, but it has not been a theme. And I don't necessarily know if it was caused by the drug. It's nothing like we see with other chemotherapy. I think that it'll take more patients so that we can get a sense of whether there's a small group that will have GI issues, but I don't think it's an issue in a major sense right now. Chris Carson wrote, will the oral formulation be time release? Curious due to the use of having to wear a bag. Yeah, the oral formulation, it's really going to be about absorption. Once it gets in the blood, it seems like it's pretty stable, but it's the absorption piece that we're having to figure out. And there's a lot of drugs that are like antifungal drugs that have the same problem. And what they tell you is to take it with a fatty meal. Like you take this drug and eat it with 4 strips of bacon and 3 scrambled eggs and something, a large pile of coconut oil or something. And then the drug gets absorbed really well. And so that's a challenge with a lot of drugs, not just ours. Karen asks, is the phase 2 open now, and what date will it run? It'll run until it's full. I'm guessing at the rate we're enrolling patients, it'll be 6 to 9 months from now. That might be a good place to start to wind up. Erica and David, if you want to unmute, if there's anything you want to say, or Christy.
Yeah. Well, I'll just thank you, everybody that's come out and supported us, and thank you for all the work you're doing and continue to do. No parent should have to go through what we do and what everybody that continues to fight this disease goes through. It's pretty brutal.
Thanks for doing this presentation. I'm telling you, with all the research you do as a parent, it was never explained so well to what's really going on with Ewing sarcoma. I mean, when you first get diagnosed, you have so much information, so none of it sticks. And then doing all the research online, you try to stop because the statistics are terrible. So
you don't want to read that online. Yeah.
You don't want to, but trying to, figure out certain things. So this was explained quite well. And, we really hope that you're onto something here.
Well, it's a hope, it's a dream. When I found out about that first patient responding, it was emotional. I mean, I was just like, I got to pinch myself that this was an idea that we came up with and somebody's being benefited from this. There's a line in the Talmud that says, it's if you save one life, it's if you save the world. And, there were 2 things that people threw roadblocks in front of us. One of them was, you'll never make a drug for this fusion protein because it's impossible to drug a fusion protein like this. And the second one was, even if you did come up with a drug, you'll never get it commercialized. And, you know, I love throwing the nocturnal thing in their face. I mean, not only is it commercialized, we're on the NASDAQ. I mean, I never would, I mean, there's all sorts of reasons for that. And I know it's not our drug. I mean, it was the other drug that really drove that. The company brought another molecule in, an antibody that's been also very successful. And because of the reverse merger, the NASDAQ thing was almost as much an accident as anything else. And it helps and it doesn't. There's good things and bad things. But just the fact that people said, you'll never get this in a company, and we're on the NASDAQ, it's like, I love sharing that with those people. So anyway, yeah, I wish, you know, I like to try to train my colleagues and talk to them about communication. I think it's really important to be able to explain these things. As you said, Erica, I never would go into this kind of detail with a newly diagnosed family, but I always tell them at the beginning that we're having you sign this consent, and consent is an ongoing process. And that means that as we go along with treatment of your child or your loved one or whoever it is, you know, we'll stop from time to time and we'll go back and re-talk about, you know, where do we come from, and we'll add a little more detail. So they'll get to understand some of these things. And that's really, really important to me. And I think that is something that, you know, I'm hoping that, you know, we've got a new group of docs at Georgetown since I took over in 2018. They're wonderful, wonderful, wonderful people. And I think that, being able to explain this and share it is a real honor for me. So I'm glad you could tune in and get a chance to see that. Christy, do you want to say anything? Let me see this.
Sure. Yeah, I just want to say thank you so much, Erica and David, for everything that you do. And it's been an honor and a pleasure to watch Dr. Toretsky work over the past couple of years, and gifts like this and grants like this really do make such a great difference. And I think I saw Dr. Toretsky the day after he heard about the patient who's on the drug, and he was very excited and definitely emotional. And it's definitely something that we're all so excited about and so great to see going on. So thank you guys again so much. And thank you to everyone who joined. It's great. Always great to hear Dr. Toretsky explain things, and if you get the message out a little bit more, it always helps. So thank you so much for joining tonight, guys.
Oh, one other thing before everybody zips off. Let me, one thing I didn't get to, let me share my screen one more time. And if anybody is interested, oops, there's how you can contact me. So there's my email. I've got a busy couple weeks coming up, so I'm not promising anything right away. And I will tell everybody that I don't make recommendations to families about treatment. I will talk to oncologists, but I don't do any phone consults or remote recommendations like that, mainly for time. And there's my lab website. If you go to that website, you can find a blog post by a father whose son died of Ewing's in London. And he wrote a really interesting story about he came and interviewed me in Washington, and he wrote the whole saga down. So it's kind of a fun read, and you can find that on my lab website if anybody's interested. So, okay. Well, with that, I want to thank Erica and David and everybody for tuning in. And let's see here. I just also wanted to say one last thing to Leslie. Leslie, I wish your son well. And that's where I'll end it. Thank you, guys.
Thank you.
Thank you.
Thank you, everyone.
Bye.
Christy, can you stay on for a sec?